Hello everyone and welcome to a video that I'm making today on Sanger sequencing. Sanger sequencing is a method that we can use to be able to determine the sequence of nucleotides that are found within a specific segment of DNA. Now, it requires thermal stable DNA polymerase because we are still going to be synthesizing DNA, and it also requires a sequencing primer. Remember that this primer essentially is like acting as this primer, like RNA primer that would be in DNA replication, for us to have a double-stranded segment of DNA for our polymerase to be able to recognize, bind to, and start this polymerization process. The next thing that we're going to need is dNTPs. In our sample, we need nucleotides for us to be able to place down to be able to actually polymerize. And lastly, we need ddNTPs. Now, think of these ddNTPs and dNTPs being very similar. These are nucleotides. However, instead of having these OHs that are very useful for us to be able to make bonds between these nucleotides, right, we actually get rid of one of these OHs. You'll actually see right here, we have what we would normally call the hydroxide on the 3' carbon, right? However, on this end, we don't have that hydroxide, meaning that although we can still form a bond over here, we cannot form a bond over here. Sanger sequencing can be done two different ways. Now, the more historical method is by using 4 separate tubes. In these tubes, we're going to add our DNA polymerase, we're going to add our primers, we're going to add our dNTPs, and obviously the specific DNA that we're trying to sequence. Now, in each of these 4 separate tubes, we're going to add separately a little bit of our ddNTPs. So in one tube, we might add our ddATP, our ddCTP, our ddGTP, and then eventually into our last tube, our ddTTP. And like this, we'll have 4 separate tubes that are supposed to give us information about the position of each one of these individual nucleotides. And we're going to get into exactly how that works. Let's imagine that we're in our ddATP tube. Now, if we're in our ddATP tube and we have our DNA sample, and let's picture that this larger sequence, this is the DNA sample we're trying to sequence. Now, obviously, we can't see this with our naked eye. This is just a representation of what we're doing. So we've added our primer, and our primer is now inside that sequence, and it's bound to the sequence that we're trying to understand, the nucleotide sequence for. Well, once we've had this done, this DNA polymerase, what it's going to do is it's going to start recognizing this sequence, and it's going to want to start actually polymerizing. So in the normal case, let's say we only had dNTPs, our regular nucleotides. Well, what our DNA polymerase is going to do, it's going to find the complementary nucleotides. It's going to be taking these dNTPs that we've added to our solution and start taking them and putting them on. So for every A, we're going to find a T. For every T, we're going to find an A, so on and so forth, right? Until we finish with the entirety of this particular sequence. Well, remember that we have these ddNTPs that are missing that 3' hydroxide group. Well, as a result of this, there's going to be moments where suddenly we're looking for a nucleotide, we grab it, we put it down, and we can't seem to add the next one. What does this mean? Let's say that we have, in this particular sample, we have our ddATPs. Well, we have an A here, so we would normally put down a T. This is a normal thymine. We don't have to worry about this being any of these modified versions. However, we get to the thymine and we have to put down an A. Our DNA polymerase is looking for any nucleotide in the solution, and it's very possible it picks up one of these ddATPs. So let's say it does pick up a ddATP. Remember that 3' hydroxide group is useful for us to be able to make the bond, for us to be able to add the next nucleotide. Well, if that doesn't happen, we stop here. We don't continue. Suddenly, the length of DNA that we've just made only goes that far. We have a small segment of DNA here. In the same solution, but in the next DNA molecule over, let's say we did add that T. And we added an A, and just by luck, our DNA polymerase happened to pick up one of the A's that is a regular nucleotide. It's not the dideoxy version. It's not the version without the 3' hydroxide. Well, it's perfectly fine then. We can continue adding our next nucleotide. So we can add our next C, we can add our next G, we can add our next T. But suddenly, we have to add another A. If we add this next A, again, we run the risk of picking up one of these ddATPs. Let's say in this particular case we did, right? We picked up a dideoxy ATP. And as a result of that, suddenly we don't have the ability to add the next nucleotide over. So this polymerization ends here, and that's it. And the next one over, let's say we do this process again. So we have our T, we have our A, we have our C, our G, our T, our A, we continue on C, G, E, And suddenly we're again facing this problem. We have our A, and we could have picked up a regular A. We could have picked up a D ATP, one that does have that 3' hydroxide group. However, in this case, let's say we did pick up again our DD ATP, it's going to stop there. Once you have this solution, think of this as being a solution filled with millions of copies of DNA, and so this is happening millions of times. And as a result of that, suddenly we're going to have so many different molecules of DNA, and all of them are going to be slightly different sizes. There's a technique we can use that can let us know the size of each of these molecules, and if we know the size of the molecule, we then know where each of the A's go. So we can use gel electrophoresis. Now, if you're not confident in your understanding of gel electrophoresis, feel free to look at my other video on gel electrophoresis where we talk about the general understanding of what this process does. Let's get into it. So let's say we load our wells and we load the solution that had our DD ATPs here, our DD TTPs here, our DD CTPs here, and our DD GTPs here. Well, anytime that we see a band, this lets us know, oh, that's the nucleotide that we're working with. So if we have a molecule that was a molecule of DNA that's only, let's say, 4 nucleotides long, right? We might get a band somewhere here. And the next one might be 5 nucleotides long, and we'll get a band here. And the next one is 6 nucleotides long, and we might get a band here. And the next molecule is 7 nucleotides long, and we get a band here. And suddenly these bands start giving us information, letting us know what our sequence of DNA is, because we can look at each one of these separate solutions, put them down a well of our gel electrophoresis, and actually see what nucleotide goes where based on the mass or the size of these molecules of DNA. So like this, we would see that our first nucleotide is an A, our next nucleotide is a T, our next nucleotide is a C, our next nucleotide is a G, our next nucleotide is a T, our next nucleotide is an A, so on and so forth. And like this, we know this is our beginning, and this is our end, and suddenly we've been able to figure out the sequence of this particular segment of DNA. And that's how old methods of Sanger sequencing used to work. As time has progressed, Sanger sequencing has also changed though. Things have improved. So now, instead of doing our 4 separate tubes, what we do now is use one tube. And we put all of our 4 ddNTPs in that same tube. We put our regular NTPs, we put our regular primers, we put our regular DNA polymerase. That's thermal stable, by the way. Now, once we have all of that in our new tube, we have to make sure that there's one modification. Now, these ddNTPs, the way that we'll be able to determine what goes where now is suddenly by fluorescent tags. Every time we have an ATP, if we shoot a light at it, suddenly it'll turn red. It'll glow red. Our CTPs, it'll glow green. Our GTPs, it'll glow yellow. And our TTPs are going to glow blue. Now, this isn't going to be for every single nucleotide, but just for these dideoxynucleotides. And mind you, these colors can be changed. It all depends on the way that you design your particular experiment. Now, once we have all of these in here, what we can then do is work with this same kind of process, work with the same kind of gel electrophoresis, except just slightly different. Instead of doing it through our regular gel like normal, we're going to put it through something called a capillary tube. And this is essentially doing that gel electrophoresis, but all in one tube. And as things come across that tube very, very slowly, we'll shine a laser at it. And every time that we see a little red light, that lets us know, oh, that's an ATP right there. For sure, in that segment, we have an A. And the next time that something crosses over that tube, we'll shine a light through that tube, and if it turns green, suddenly that green means we have a CTP. And if another nucleotide follows through that tube, and we shine a light through it and it turns yellow, we have a GTP. And the next time that it comes through, if it turns blue, we know we have a TTP. And so suddenly, based off the color of light, as opposed to having these 4 separate wells on the gel, based off of these colors of light, we can actually see that we have a specific sequence of nucleotides. And now this information is read using something called an electropherogram. And this electropherogram essentially shows us the order at which these colors came through that tube. And suddenly we can then say, oh, I have this blue. I know for sure if I look back at my old legend, if I have blue, I have a TTP. And so I can say that I have a T here. And so on and so forth. If you follow your legend, you can eventually figure out the sequence of nucleotides you're working with. And like that, we've then updated Sanger sequencing to be able to be applied in a slightly different way. I hope that this helps. If you have any questions, comments, or concerns, don't hesitate to reach out. Best of luck.